Hi everyone, my name is Sid, and today I'm going to be talking to you about connectome-based spreading of grey matter changes across the psychosis spectrum. I have no disclosures. So I think one of the most common biological findings in psychosis is the loss of brain tissue. And aside from a few post-mortem and pneumoencephalographic studies, we really started to quantify this in the 70s using CT scans, which showed increased ventricular size in patients. And in the late 80s, there was enough studies using MRI and CT to show that when you conduct meta-analyses, you see changes in frontal temporal and medial temporal regions, and preferentially. In the 2000s, the technological advances really allowed us to pinpoint changes which seem to be, at least at a group average level, all over the brain. And today we have massive sample sizes of thousands of patients and can even roughly stage psychotic illnesses and relate brain alterations to these stages of less severe to most severe. And what I'm trying to get at here is that we've been mapping the brain for a long time in psychosis, and we really need to move on to understanding the mechanistic causes of these brain changes, or really trying to model the cause of what causes these brain changes. And there are many levels to that, and I'll be talking about today modeling the process which causes the spatial pattern that we see often in psychosis of anatomical brain changes. One recent and I think important insight from neuroimaging and neurological conditions is that brain volume loss is not randomly distributed over the brain, but is concentrated on specific functional and structural systems. And I think this figure is a powerful demonstration of this, where the spatial pattern of atrophy in Alzheimer's, frontotemporal dementia, semantic dementia, and other diseases matches certain large-scale functional and structural networks derived from healthy populations. And one explanation for this is that the complex web of axonal fibers that connect our brain regions may be a conduit for the pathological spread of an illness-related process or a medication effect. For instance, an illness or medication process originating in one area can sequentially spread to other areas via the white matter connections. And this can happen via multiple mechanisms. Alternatively, these regions can have a shared vulnerability, and that would also show this sort of pattern. There are a few reasons we are starting to think that these types of network constraints also apply to psychosis-related brain changes, and even hints of a spreading process. So atrophy, or volume loss rather, in psychotic illness is tightly correlated between spatially distributed regions, so structural covariance is high. It's also correlated with adjacent white matter and connecting tracks, and this is cortical thinning is correlated with changes in the connecting white matter. And the loss of thickness, so cortical thinning again, is correlated with normative connectome organization. What I mean by that is the more axonal connections a region has, the greater gray matter volume loss or thinning it shows. And we thought this was enough evidence to start trying to model the mechanisms of spread of volume loss using kind of an extensive sampling approach. So to do this, we applied a very simple mathematical model that was recently used in the literature. And first I'll describe the ingredients of this model. We want to get really good group level estimates of volume differences in multiple samples of patients with varying severity of illness. And here we used 4 studies comprising patients with unmedicated first episode psychosis, early psychosis, and 2 samples with established schizophrenia. And we used a technique known as deformation-based morphometry to generate kind of unthresholded effect size maps, which is what I'm showing here, with positive values at the voxel level shown in red signifying loss, volume loss, and negative values shown in blue signifying volume expansion compared to a matched healthy control group for each of these samples. We then parcellated the brain into 332 regions, and where we have different estimates of volume change as the mean of all voxels belonging to that region. So to implement this model, we also need solid estimates of the pattern and strength of functional coupling or connectivity between regions in a representative healthy brain. And using a large, completely independent sample of 300 adults, we used diffusion and functional MRI to get consensus estimates of the strength of structural and functional coupling between each pair of these same 332 regions. So what we have so far for these 332 regions, we have different estimates of volume change for each of the samples, as well as representative or normative structural and functional coupling strength between each pair of regions. And then we implemented 3 variations of this neighborhood deformation model, which I'll take you through now. So if there is a spreading process occurring, it could be hypothesized that the volume change in one region would be correlated with the average volume change of its connected neighboring regions. Likewise, if you take this region, you should be able to get out its volume change by the change in its 3 structurally connected neighbors here. And this was simply the first model. The second model, we took into account the fact that the structural connections between regions vary in their strength, myelination, axon number, multiple features. It's a weighted measure. So for a given region, the volume change in each of its connected neighbors was multiplied by the strength of structural coupling between the regions. So regions with a little change but a strong connection were upweighted, and regions with lots of change and a weak connection were downweighted, and this was the second model. The third and final model followed the exact same form, except this time we multiplied the volume by the functional coupling from the value from the functional connectivity matrix rather than the structural connectivity matrix. And to test each model's performance for each data set, we computed a Pearson correlation between volume change, observed volume change in a given region, and predicted volume change according to one of these 3 models. And we tested this performance against 3 benchmark null models, which I won't go into now, but I can expand if you're interested in person. So going straight to the results, what I'm showing here is just the regional level volume estimates mapped onto the parcellated brain. So same as the voxel images in the previous slides, just projected onto a surface. So focusing on the cross-sectional differences for each study, on the y-axis is the correlation between observed and model predicted volume differences. And on the x-axis, I'll present the 3 models. And for each of the 3 models, the null distributions of the correlations are given with 3 colors. And the observed correlation, which is what we care about, is highlighted in a black dot and a red circle if it beats 95% of null distribution and is significant. And what we find is that in our first episode sample is that the model that gives us the strongest correlation is the one where the volume of the neighbors was weighted by the strength of structural connectivity. And this was almost 0.5 and much better than that of the other 2 models, which were not weighted or weighted by functional connectivity. And what was really interesting is we found the exact same pattern of results for each of the other samples. So illness-related volume change in a brain region is strongly correlated with differences in structurally connected neighboring regions, and the strength of these connections really matters. And we were lucky enough to have longitudinal data from a triple-blind, placebo-controlled ICT of the first episode patients in the previous slide, which allowed us to parse out illness and medication results, effects. And here we're indexing change in volume compared to change in volume in healthy controls rather than volume differences. And we had this data at 3 and 12 months. And what's really interesting is we found the same pattern of results again when looking at longitudinal illness and medication-related changes at 3 and 12 months. So when the neighborhood deformation model was weighted by the strength of structural connections between 2 regions, we get strong and significant correlations which beat all non-models, both for volume differences across multiple samples, as well as longitudinal volume change in an early psychosis sample, as well as medication-related change, suggesting this is a kind of general feature of brain changes. And what this effect, I think, is telling us, and there's 2 main theories which we think is potentially driving this effect. Either there's a shared vulnerability, meaning that regions with shared morphological, molecular, genetic properties share stronger axonal connections. So an illness process targeting, say, a cell type or tissue property will result in this node-neighbor coupling, and there's some evidence for that. Or there's a spreading process occurring where a pathology starting in one region spreads to structurally connected regions over time. and we're investigating both possibilities, which are not mutually exclusive. But today I'm going to show you some results for some simulation modeling where we directly test the spreading hypothesis. To stimulate the spread of pathology and identify potential epicenters where pathology may start, we use the network diffusion model, which is essentially the classic heat diffusion equation unfolded on a normative structural connectome. And here we sequentially seed pathology at each brain region and allow it to spread along the connections. And for each seed at each estimated time point, or each simulated time point rather, we see what correlates best with our observed abnormality or t-statistic map from each sample, which we're using in the previous analyses as well. And we evaluate the strength of correlation between the simulated and observed, retaining only the maximum correlation over the simulated time for each seed, or Rmax. And we can create a null distribution of Rmax values using rewired structural connectomes, which preserve low-order spatial properties. And using this for each dataset, we can hopefully detect epicenters from which, if we start a spreading process, we can recapitulate or recapture the observed pattern of volume loss that is observed in these samples. And using this model, we find that during the first episode of psychosis, potential epicenters of illness lie in posterior visual and temporal as well as hippocampal regions, with primarily posterior cingulate and hippocampal regions emerging as epicenters as in earlier illness stages, and in established schizophrenia, we see largely temporal and hippocampal regions. And I think two things stick out here. The medial temporal lobe and the anterior hippocampus specifically is implicated as a potential epicenter across all illness stages. And these are independent samples. And overall, this does seem to be a posterior to medial temporal shift as the illness progresses. So to kind of summarize what I've shown today, illness and antipsychotic medication-related volume changes in a region are strongly coupled with changes in structurally connected regions. This region-neighbor relationship is modulated by the strength of structural connectivity between regions, and epicenters of illness progress from posterior to temporal regions, and the anterior hippocampus seems to be implicated across all illness stages. And that seems to align with some animal model as well as multiple theories of the onset of psychosis. And I would like to thank Alex Fornito, who was my advisor for this work, as well as the clinical team at Origin, who provided a lot of the data that we used here, and my current lab, the Holmes lab at Yale. So there's a preprint out of this work, which you can check out via that QR code, or you can come chat to me at poster 212 or 1775, and I'm happy to chat about this work. Thank you for listening.